In this video, we are going to be talking about the synapsis of chromosomes during meiosis and possible chromosomal aberrations. We are going to discuss two individuals. The first individual has the chromosomes MLI centromere JKHG and GHI centromere JKLM. In order to have the chromosomes match up as much as possible, we will flip one of them. Now we have MLI centromere JKHG and MLKJ centromere IHG. Looking at these two chromosomes, we can see that the I centromere JK and the KJ centromere I portions are flipped 180 degrees, but the rest matches up. This is called a pericentric inversion because the portion that is inverted contains a centromere. If it did not contain the centromere, it would have been a paracentric inversion. To determine if the chromatids created during meiosis will produce viable gametes, we have to draw an inversion loop. The top two lines in the inversion loop are the first chromosome, and the bottom two lines are the second chromosome. A loop has to be formed in order for them to match up as much as possible. Next, I will determine the chromatids and if they will produce a viable gamete or not. I will start by following each of the chromatid lines with a different color. There is a crossover between the J and K region, which is noted by the X created by the blue and purple chromatid lines. We can list the chromatids created by synapsis during meiosis for this individual based on the regions that they cross. The first chromatid is viable because it contains every region and the centromere. The second chromatid is not viable because it contains two sets of the M and L region and no H or G region. The third chromatid is viable because it contains every region and the centromere. And the fourth chromatid is not viable because it contains two sets of the H and G region and no M or L region. One way to understand an inversion loop is to think of it as a roller coaster that has 4 different tracks and the riders don't know which track they will be riding on. The first track has a big drop-off and the riders get to experience the roller coaster from start to finish, which makes them happy, so it is considered a successful or viable ride. The second track starts off good, but then it has a crossover which leads it back to where it started. So the riders are not happy because they do not get to experience the full ride, and so it is not considered a successful or viable ride. The third track on this roller coaster has a super fun loop-de-loop, which the riders really enjoy, and they get to experience the roller coaster from start to finish, so it's considered a successful ride. The fourth track on the roller coaster starts off good, but then it has a crossover which leads it back to where it started, so the riders are not happy since they didn't get to experience the full ride, and so it is not considered a successful or viable ride. Individual 2 has the following chromosomes. In order to have the chromosomes match up as much as possible, we will need to flip one of them. Now we have 2 completely matching chromosomes. Because there are no aberrations, we know that the synapsis will result in straight chromatids with a crossover between regions J and K. The chromatids that are resulting from the synapsis are listed, and they are all viable because they contain one of each region and the centromere. To better understand this concept, we can think of this as a 100-meter track race between 4 schools. All of the athletes compete the race from start to finish. The second runner tries to cross the path of the third to get ahead, but they all end up completing the race, so they are all successful or viable. This individual is considered more fit because it will produce more viable gametes than the other individual.